This is Dr. Mobeen Sayed with one more episode of Long Story Short with Dr. Bean from the FLCCC platform. We'll have a primer about what are the ANA, what are the anti-nuclear antibodies, how are they produced, and what parts of a cell do they attack and cause the problem, and why are they produced. So let's start our discussion. Now I want to go over what are ANA, just a primer. What are ANA, how are they produced, or why are they produced? And then how do these antibodies get into our cells and attack their target antigens? Now remember, here we're going to use the word antigen for our normal proteins. When our immune system looks at our body, everything is an antigen, everything is an enemy. Immune system has to learn to ignore its own tissue. But sometimes it forgets to ignore, and then the autoimmune diseases start. So let's start the discussion. So here we're going to discuss anti-nuclear antibodies. So the first question, are anti-nuclear antibodies? What are these? The answer, these antibodies, as the name is, anti-nuclear antibodies. These antibodies are antibodies against various parts, especially the nuclear parts of the cell, the parts inside the cell nucleus. However, it is not necessary that anti-nuclear antibodies only target antigens inside the nucleus. They can actually target antigens that are in the cytoplasm. And the example are, for example, RNP, ribosomal ribonuclear protein antigen complex, or amino-acyl transfer RNA synthetase. And synthetase means something that makes it. So transfer RNA is a kind of RNA that is present in our cytoplasm, and it helps the ribosomes to make proteins from messenger RNA. So this protein that does amino-acyl transfer RNA function or synthesis, that can be a target of anti-nuclear antibodies. So the message that ANA antibodies are not only targeted at the proteins that are inside the nucleus or even the nucleopores or even the surface of the cell, but also in the cytoplasm. So what are the kind of antibodies? Let's look at them. For example, we can have anti-histone antibodies. And histones are these octamers, or there are proteins, small spherical proteins that make a complex around which the DNA is woven, it is wrapped. So our DNA is such a long string that if it is just present inside the cell, it will be all tangled up and it will become a mess. And we have to open up the DNA every so often, almost every minute, every second, to open various genes of it. We read them, we make copies of them into messenger RNA that helps make us protein, and the cellular functions go on. So it has to be really tightly organized so that we can open a book page, use that recipe, close the book, open another page. So how does our cell handle that? We make histones that are proteins around which the DNA is wrapped. So there are antibodies that start targeting these histones. So imagine if the histones are targeted and their function is disrupted, then the DNA function will be disrupted and the whole cell can actually be in a really bad state. Then there are antibodies, anti-nuclear antibodies, that, for example, start targeting double-stranded DNA, so the DNA pieces themselves. So this happens, for example, SLE. Anti-histone antibodies are usually seen, or more likely to be seen, with drug-induced lupus. Then, as I said, we have amino-acyl transfer RNA synthetase. So let's go here. When a cell is apoptoting, when a cell is dying, at that time, on the surface of cell, some antigens or some proteins become expressed. A dying cell's behavior is different from a normal cell's behavior. So as the cell is breaking down and dying, there are some proteins that appear on its surface. There are two important proteins that are interesting. One is Rho and the other one is La. So these antigens, when they appear, our immune system can go mad at them to say, who are you? I have not seen you here before. And the antibodies can be produced against them. So these will be anti-Rho or Sjogren's syndrome A, SSA or anti-Rho antibodies, or anti-La or Sjogren's syndrome B antibodies. So as you can see, it's not necessary that these are all sitting inside. And another thing that you may be observing, apoptosis is a very important component of our body's function that can give rise to autoimmune diseases if it is sustained. And I'll explain that a little more. Just look at a variety of nuclear antibodies. Then we have anti-centromere antibodies. So we know that centromeres are part of the chromosome in the center, and there can be antibodies directed to the centromeres themselves. Then this is the one that I said I'll discuss later. So inside a nucleus, there are tiny proteins whose job is that when messenger RNA is formed. So think about it with me for a second. Imagine we have DNA and we need to open up. DNA is a book of recipes for various functions and proteins of our body. Functions, proteins mainly. So imagine if we want to open up a specific page and we want to copy the recipe, that copying of the recipe is going to form messenger RNA, or the copy is called messenger RNA. When the copy is initially made, when the messenger RNA is made inside the nucleus, that messenger RNA still needs further processing. Some introns or some parts of the messenger RNA needs to be removed. It needs to be capped. A tail needs to be attached. So for that, there are processing units that live in the nucleus that help process the RNA. One set of units is called ribonucleoproteins. As the name suggests, ribonucleoproteins. So it is nuclear component plus attached with the polypeptides or proteins. These complexes form with other proteins. They form what we say spliceosomes. Spliceosomes, as the name says, splicing. So U1RNP is a spliceosome. If you see here, this is the ribonucleoprotein, and with that attached are proteins or polypeptides. So this is ribonucleo part, and then here are proteins. So this is a ribonucleoprotein. And if you see here, this little tiny thing is a newly born RNA, messenger RNA. And this will be soon spliced up and gotten ready to have a cap attached to it and a tail attached to it, and the introns will be removed. And antinuclear antibodies can form against these complexes. Imagine this, that now when the nuclear antibodies are attached to this complex, this machine, this processing unit for messenger RNA, then the messenger RNA production will be disrupted and the cell function will be disrupted. So for example, anti-Smith antibodies, which are for the SLE, then here anti-nRNP or anti-U1RNP that are for SLE and connective tissue disorders. Similarly, there are many more. I'm just giving some examples. Similarly, there are some antibodies that, for example, are against topoisomerases. Topoisomerases are the enzymes, again present inside the nucleus, that help build or open up a gene and use it and then close it back in. And then the DNA replication. These are the enzymes that take part in such functions. And now we have antibodies against them as well. For example, SCL, scleroderma 70 antibodies. So once again, the basic target is the nucleus or genetic material related targets. But there are some that are in the cytoplasm, some on the cell surface as well. So these are some examples. Now the question are, why do we end up with these antibodies? And this is a very important question, and the answer would help us understand long COVID. It would also help us understand that when there is persistent inflammation, how that can give rise to ANA, antinuclear antibodies. And then presence of ANA would further increase inflammation. So we have to break this cycle to help the situation. So first of all, antinuclear antibodies sometimes are produced when there is defective apoptotic genes in a T cell. So the function of a T cell, helper T cell for example, is to activate the B cells or cytotoxic T cell to take care. If B cells are activated, they will make antibodies. Now imagine an autoreactive T cell. When the T cells go to our thymus, they are tested to see if they are going to react to our own body. And if they do, we kill them. We ask them to perform apoptosis. Apoptosis means dying by self. We tell them to apoptose and just go away. But if their apoptosis machinery of a T cell is not working correctly, then even when we tell that cell that, hey man, you are autoreactive, you're going to cause damage to our body, you should die, it just lets it not die. And now that autoreactive T cell would come out in the tissues and would start reacting to our body. And that would cause, if it reacts against the nuclear components, then that would cause ANA. It can be other kind of reactions too. So that is one way antinuclear antibodies can be produced. Incorrect T cells attacking our own body. Then look at this one. Prolonged apoptosis. This is very important. So the cells that become old and are cleared away by, let's say, other cells, macrophages for example, usually they do not trigger the immune system. So usually apoptosis of a regular physiological kind does not trigger the immune system. And you could say, why not? And one general answer will be that when normal natural apoptosis or senescence of a cell and cell death occurs, the proteins inside the cell are not damaged. They are not de-shaped. They are not deformed. It's just that the cell has reached its age. Plus, those proteins are released within the macrophages most of the time, and macrophage would not become activated because it knows I'm clearing out these cells. However, if apoptosis occurs, now this is a delicate point, if apoptosis occurs because there was a stress on the cell, for example, oxidants, reactive oxygen species, or other inflammatory markers that are causing the cellular functions to be disrupted, or remember the cytoskeletal disruption of the cell, the skeleton of the cell that becomes disrupted, or other chemical substances, or acidosis, or lack of carbon dioxide, or presence of carbon monoxide, or a virus, or other things. If there is a stress, then cell would try to kill itself as well if it cannot handle that stress. Such cell, think about it with me, such cell, when it would break down, it would express proteins that may not be entirely in their normal natural shape. Why? Because the cell died of a stress. And depending upon the kind of stress, the proteins may have been deformed. That is why the cell died. And those deformed protein, although they are our protein, they still are looked at by the immune system to say, this seems foreign, this seems unknown to us, and the immune system starts reacting to the components of this cell, including normal nuclear components too, and that would create ANAs. Then another problem with the persistent phagocytosis or inflammation. So imagine somebody has COVID, and after that there is long COVID, and there is continuous inflammation, which is causing continuous death of cells and abnormal deaths. Guess what happens? In addition to this, and this is not just COVID, many inflammatory diseases would do this, the reactive oxygen species are present and they are deforming the proteins. But here is a delicate point. Even after the cell has died, so imagine a cell that has died and the guts are opened and spilled out. These peanut-looking things are actually the cell's, various organelle and proteins, and these strings are the DNAs and RNAs, and these are histones and nucleus and the cell membrane proteins. Even when the cell is dead, just like vultures, the reactive oxygen species are still attacking this dead cell. And as they are attacking right in front of the immune system cell's eyes, these reactive oxygen species will be deforming these proteins. And as these proteins are deformed, their shape changes, and to our immune system cells, these look foreign, these look antigenic, these look dangerous, and the immune system would start reacting. So please realize, persistent, prolonged cell deaths because of inflammation have a chance of giving rise to autoimmune diseases because the proteins may already be denatured or still being denatured because of the presence of reactive oxygen species. This is like fireworks in a dead cell, and that firework would cause damage. Now, could this inflammation be because this is happening or because antinuclear antibodies were present before? Yes. Could these be after? Yes. Meaning, could there be another reason for the cell death? And then the ANA are produced, and then the ANA start attacking our cells as well, and further inflammation occurs, and further cell death occurs, and this cycle continues. So a rheumatologist or a doctor have to be trying to break this cycle and give rest to immune system so these cells can actually go away and be cleared out so that immune system can start taking a break. Okay, so then defective clearance. And I'll give you an example of a defective clearance. One example is, for example, macrophages, neutrophils, dendritic cells. They help pick up debris, macrophages especially, and recycle it and clear it. Sometimes that clearing mechanism isn't correct. So these dead carcasses of the cell, these apoptotic cells, these broken cells, necrosed cells are sitting there lying in the tissue, acting as antigens, or the proteins acting as antigens, but they're not cleared out correctly. Maybe they are so large in number, the destroyed cells, that our clearing system is overwhelmed. We will make more clearing cells and they would eventually clear it, but during that time, that litter present, the necrosed tissue present, may look to immune system as an enemy. So then an example of defective clearance. In our body, there is a protein called, this green little monster here, a protein called serum amyloid P component, or SAP. The serum amyloid P component does many functions. One of the function is that when a cell is dying, this serum amyloid component will come near the histones with the DNA wrapped on it. And what it will do is this. In this diagram, if you see, this little histone structure has this little pole-like structure here. This is histone 1. So what this protein actually does is it separates the histone 1 from the remaining histones. That kind of unravels this little organizing unit, binder for the DNA. As soon as that is unraveled, then this protein can bind with the chromatin and then make it solubilized and circulate in the circulation to take it to, for example, spleen or other macrophages who can clear it out. Important thing is that we need to solubilize the chromatin or the DNA pieces that are present in the tissue and circulation, and one such protein that helps with that is the amyloid P component. Now, if the amyloid P component or SAP protein itself is damaged, then it cannot perform the splitting of the H1 from the remaining histone, or the nucleosome as we call it. And that causes the DNA and histone and those pieces to stay out there in the tissues and circulation, which will then be looked at by the immune system as antigens, and immune system would develop antinuclear antibodies. These antinuclear antibodies against these little broken systems are fine. But the problem is these antibodies are going to go to healthy cells and enter those cells and go and attack the antigens. So that brings us to the last part of our discussion, and that is, you could say, antibodies usually do not enter cells. For example, Listeria monocytogenes. It is a pathogen that causes listeriosis, and Listeria monocytogenes is called a facultative intracellular pathogen, meaning it can grow outside a cell, but it can grow inside a cell as well. And one of the virulence factor or advantage of that bacteria for bacteria and harmful for us is the capability of hiding inside the cell that prevents the antibodies against it from harming it, because antibodies are standing outside the cell and this guy is hiding inside. So generally we can say that antibodies do not enter inside the cells. However, anti-nuclear antibodies have been now observed many times in experiments that these can actually come in contact with the cell surface. Cell would actually endocytose them. They are able to trigger production of small carrier vesicles which would bring them in, and then they are liberated inside the cell and then they wreak havoc there. So these antibodies can actually enter the cell, healthy normal cell, enter there, attach to the components in the cytoplasm, of course attach to the components outside of the cell much easier, then even get into the nucleus and attach to various things that I mentioned above. So I hope that this clarifies how the anti-nuclear antibodies are produced, raises a question in your mind to say, do they come first, or inflammation comes first, or is there a cycle? My answer is there is a cycle, and in various people, either apoptosis and inflammation would come first because of some disease, and then ANA are produced, or maybe ANA were produced first because of some genetic abnormality, and then the inflammation, continuous inflammation or chronic inflammation occurred. I hope that this is also clear that what kind of cellular target antigens are their target for these antibodies, and also that when these antibodies are targeting their antigens, what kind of cellular disruptions can occur, functional disruption and even cell death. And also the role of abnormal apoptosis. Normal apoptosis doesn't do this. As I said before, the proteins seem to be healthy and normal. But abnormal apoptosis with denatured protein or denaturing proteins can give rise to antinuclear antibodies. So I hope that you enjoyed this lecture. Here we actually have two separate topics, but they were related, so I discussed them together. Thank you very much. I would see you next time. Bye for now.